So this is a really, really fascinating topic. And if you think lipids below the brain are complicated, get up into the central nervous system. That's a real trip. And so in general, what's good for the heart is good for the brain. Okay. So exercise is associated with a 45% decrease in Alzheimer's disease. Like I say, if exercise was a drug, it'd be a miracle pill. And so that's wonderful. And then people with the highest cardiorespiratory fitness have the lowest incidence of dementia at a population level. Okay, well, that makes sense. And then if we look on the other side of the equation, if people have established cardiovascular disease in midlife, that's associated with a 29% increased risk in Alzheimer's disease from the UK Biobank. And then also, there is a signal for increased Alzheimer's disease if people have elevated ApoB in midlife. And ApoB is a measure of potentially atherogenic particles. And it's a much better measure of the risk in your lipid neighborhood versus just an LDL cholesterol. When people have normal LDL cholesterol, but elevated ApoB, that person still has increased risk of cardiovascular disease. And those people do have an increased risk of Alzheimer's disease from the UK Biobank as well. Additionally, midlife obesity, you have nearly a 4-fold risk of Alzheimer's disease. And, and obesity is more than BMI, it's more body composition because, every NFL player is obese. And yeah, I'm not worried about that guy, am I? So anyway, but really, obesity in midlife, you're probably retired at that point. And, that's probably not great. And, and so anyway, so 4-fold risk of Alzheimer's disease. And additionally, diabetes, 56% increased risk of Alzheimer's disease. And then people with the highest CAC scores in the MESA consortium have the highest incidence of dementia as well. And then control your insulin, control your life. People with hyperinsulinemia have an over 2-fold risk of dementia as well. So all of these things make sense, right? And most of these things are related to more the home security system element. But if there's been a break-in, we see increased signal for Alzheimer's disease or other types of dementia as well. That said, the lipid neighborhood is a lot more complicated when it comes to the brain because what's going on with cholesterol transport below the brain is kept separate from what's going on in the brain. Now, 20 to 25% of total body cholesterol is in the brain, but the half-life of cholesterol in the brain is 6 months to 5 years. Whoa, that's a long time. And so conversely, below the brain, the half-life of cholesterol is a few days, and the plasma residence time, even on an HDL particle, if they linger a while, maybe 5 days, So it really is recycled with a greater frequency than it is in the brain. However, in the brain, instead of the ApoB lipoproteins being the primary mailman, it's actually ApoE HDL-like particles that are the primary mailman in the brain. And how it's supposed to work is the astrocyte, which is a star-shaped glial cell, and glial is like a support cell for the neuron. Those are the guys that actually synthesize cholesterol. And then that cholesterol is transported via these ApoE particles to the neuron, so the neuron can use the cholesterol for its whole host of synaptic transmission and all the other things that the neuron uses cholesterol for. And the reason why this is important is because cholesterol is very, very energetically expensive to make. It requires 36 ATP in order to synthesize a molecule of cholesterol. That's a lot. And the neuron's got all sorts of other stuff to do, right? The neuron's running the show. The neuron's running the body. And he's kind of like the CEO of the body. And so the last thing the neuron needs to do is, okay, hold on, guys. Let me synthesize all this cholesterol. No, that's grunt work. That's what the astrocyte's there for. And so ideally, the astrocyte makes the cholesterol and gives it to the ApoE particle. And the ApoE HDL-like, and it's called HDL-like because it's in the same density range, but it's not really an HDL. All these things have similar names, but their functions are different in the brain. And that's why it's like, oh my word, I don't even know if I want to be friends with this guy anymore when you start talking about topics that are this complicated. But anyway, but really the astrocyte synthesizes cholesterol and passes it off to the ApoE HDL-like particle. And ideally, the ApoE particle transports it to the neuron, and the neuron uses it, and then we're all good. The problem is people with something called ApoE4, and so I say ApoE Thor sometimes because Chris Hemsworth, who plays Thor in the Marvel movies, has 2 copies of this ApoE4, which is the number one that we currently have identified genetic risk factor for late onset Alzheimer's disease. And so people that have 1 copy of ApoE4 tend to have a 2 to 3-fold risk. and people who have 2 copies of ApoE4, maybe 8 to 12-fold risk of Alzheimer's disease. But it's not deterministic. Just because you have one of these doesn't mean that you're going to get Alzheimer's. It just means you might have less margin for error. So anyway, so basically though, these ApoE4, if you have ApoE4, then they're like easily distractible members of the office. And so when you get the cholesterol memo from the astrocyte, instead of actually delivering it to the neuron, the ApoE4 particles are easily distracted. And it's like, oh, what's that? And then they leave messes in places in your brain. And then when cholesterol accumulates in places it doesn't belong, it's very, very pro-inflammatory. And what happens is the microglia, which are the immune cells in your brain, get annoyed with the ApoE4 distractible members of the office. And they're like, this is the worst. And so they try to clean up the mess and mount an inflammatory response in the brain. And that's not very good. And what happens too is the neuron will start synthesizing its own ApoE and its own cholesterol. But when the CEO is doing things that the support staff should be doing, you're asking for burnout. And so that's why in the Alzheimer's disease brain, we really do see consistently 3 cholesterol disturbances. We see defective cholesterol transport, and that's the easily distractible ApoE4 guys. We see impaired cholesterol elimination. And so basically messes are accumulating and then they're not getting taken care of. And there is a way to eliminate excess cholesterol from the neuron. It's called 24S hydroxycholesterol, which is a separate topic. But if we are unable to eradicate the cholesterol that's accumulating there, then we're asking for problems. And then the third hallmark we see is decreased cholesterol synthesis, because it actually negatively feeds back on overall cholesterol synthesis in the brain. And so once again, we see defective cholesterol transport, impaired cholesterol elimination, and then decreased cholesterol synthesis consistently in people with Alzheimer's disease. And so really when you're a hammer, everything looks like a nail, but I think Alzheimer's disease is a lipid disorder. And I think that really the problems we see with amyloid beta accumulation and hyperphosphorylated tau and the neurofibrillary tangles and subsequent neurodegeneration that we see in Alzheimer's disease, I honestly think is a consequence of impaired substrate utilization, specifically impaired glucose utilization across the lifespan, which leads to the lipid disturbances that we see in Alzheimer's disease. And then we get caught in this nasty cascade where we have these abnormal protein signatures accumulating as well that further perpetuate that disease process. And so anyway, there's all this to say, it's a trip, definitely. But I do believe that there's hope on the horizon. I believe that as research is improving, and especially as biomarkers are improving, because people aren't just signing up to get brain biopsies or get cerebrospinal fluid taps, which is, how crazy is that? And so it's hard sometimes to know what's going on in the brain when it comes to metabolism and when it comes to cholesterol homeostasis. However, we're getting better at having these blood-based biomarkers like p-tau217, like amyloid beta 4240 subfractionations, and then also having a better understanding of some of the specific things that we can understand about cholesterol regulation, such as desmosterol levels, and potentially if we're able to have a better understanding of things like PCSK9 in the brain. And so I actually am very optimistic that we're going to have an individualized regimen for people who we have identified in the early stages of Alzheimer's disease or subclinical Alzheimer's disease or mild cognitive impairment, so that we can actually act in a specific, nuanced way to help them not go down that terrible, terrible road of neurodegeneration. Because there's a big difference between living and existing. My grandmother passed away from complications related to Alzheimer's disease, and it breaks your heart. And you don't want that for anybody. And all of our lives have been negatively touched by dementia, and we don't want that for anybody. So anyway, I'm actually really optimistic